This is Bob. Say hi, Bob. Bob is an apolipoprotein B protein. We are going to see his journey to becoming a fully functional protein. Here are the parts of the cell we are going to be exploring, known as the endomembrane system. We start inside the nucleus. This is a strand of DNA, and right there is an enzyme called RNA polymerase. It is reading this section of the strand, known as a gene, which is a unit of DNA that holds the instructions for building specific proteins. It is doing a process known as transcription, making a new single strand called mRNA, or messenger RNA, which contains instructions for protein synthesis. The next part of our journey is the nuclear envelope, which is the membrane that surrounds the nucleus. The nuclear envelope is a double membrane system made up of two phospholipid bilayers. After our mRNA has matured, it can leave the nucleus through a nuclear pore, which regulate the passage of large molecules such as proteins and RNA through the nuclear envelope. Some mRNA will go to free ribosomes, but since Bob is going to be an apolipoprotein B protein, which is a secretion protein, he will be going to the rough endoplasmic reticulum, which is a part of the whole endoplasmic reticulum, which is a vast network of flattened sacs and tubules that are continuous with the nuclear envelope. They are comprised of two parts: the smooth endoplasmic reticulum, which makes lipids such as phospholipids, steroids, and oils, and also detoxifies drugs and poisons, and stores calcium ions. The other part is the rough endoplasmic reticulum, which is for synthesizing certain proteins like membrane proteins and secretion proteins like Bob. It is called rough because it is studded with ribosomes. Ribosomes consist of proteins and rRNA, which is synthesized in the nucleolus. Now our mRNA has been translated by the ribosome, and there is Bob, but he still needs to be worked on. Now part of the membrane is butted off into a transport vesicle to transport Bob. The transport vesicle is then carried across a microtubule by a motor protein called a kinesin. Our next stop is the Golgi apparatus, where Bob's vesicle enters the cyst side, which is where the Golgi apparatus receives vesicles. Proteins in the Golgi apparatus are modified and finalized for function. An example is with Bob, who's being phosphorylated, or having a phosphate group attached to it. Now Bob is finally complete and ready for export. A new transport vesicle is formed. Bob's final destination is the plasma membrane, which is the barrier around the cell that regulates what's coming in and out to keep the ideal environment for function, letting essential nutrients in and toxins out. The vesicle fuses with the plasma membrane, and Bob is out of the cell to do what he was made for. But what if Bob was destined to be a digestive enzyme? Meet Billy. He had gone through the same process so far as Bob, except for he didn't go to the plasma membrane. He is a lysosomal enzyme known as a cathepsin. His vesicle is known as a lysosome, which budded off the Golgi apparatus, made of its membrane. They can fuse with food vacuoles to digest food, and vesicles containing damaged organelles to recycle cell parts. The end.